Hello children, so today we are going to see the part 7 of molecular basis of inheritance. So hope you must have studied the previous parts 1 to 6. So today we are going to see the DNA replication. So you know central dogma theory. In central dogma theory, so DNA it can replicate. So the replication of DNA you can call that as duplication also. So mainly the duplication of DNA it occurs during cell division. So then later the DNA transcribes to form mRNA, and the mRNA later it forms protein by means of translation. Okay. So DNA it forms the DNA, you call that as duplication. The DNA to RNA you call that as transcription, and RNA to proteins you call that as translation. So for replication it needs some replication machineries. So when does the replication start? Mainly it occurs during cell division. So before cell division the number of chromosomes found it has to be doubled. So that's why the replication occurs. For example, let us study regarding E. coli. In E. coli, for complete replication of its DNA, it occurs within 38 minutes. Average rate of polymerization, it is approximately 2000 base pairs per second. So in case during replication, if there is any mistake occurs, so that results in mutation. So mutation is sudden heritable changes. So if there is any change occurs in DNA, then the RNA formed will be wrong. When the RNA formed is wrong, the protein formed also will be wrong. So that's why it can result some problems. So it can be metabolic problems or it can be any other diseases. So that you call that as mutation. So deoxyribonucleoside triphosphate. So here it can be ATP, GTP, CTP or TTP. That is adenosine triphosphate, guanosine triphosphate, cytosine triphosphate or thymidine triphosphate. They serve two purposes, dual purposes. So one, they provide the energy for polymerization or act as substrates for polymerization. So here the replication, how does it start? So for starting of replication, the DNA it has to open. So the opening that you call that as formation, that the opening that you call that as replication fork. The place where the replication, where the fork is formed, that you call that as origin of replication. The replication fork is formed by an enzyme called helicase. So this enzyme it is helpful in the formation of replication fork. So once it is opened, then the strand means the new strands can be formed. So here you can see one is actually it has been given in two colors for identification. So here one strand you can see in blue in color, and this side also blue in color. These blue colored strands, they are old strands, that you call that as template strands. So the pink color or red color, whatever they are given, so these strands are newly formed strands. So one side you see here the DNA they have written as continuous synthesis, other side it has been written as discontinuous synthesis. So one thing you have to remember, always the newly formed DNA strand it is from 5' to 3' direction. So here it is catalyzed by the enzyme called DNA polymerase. Actually what I shown you in the previous diagram. So here this diagram you can see here the blue colored strand, that is template strand, and the pink colored strand, that is the newly formed strand. So here when you see in the case of, I already told you, the DNA polymerases it catalyzes polymerization only in one direction, 5' to 3' direction. So here, so when it has to be from 5' to 3' direction, the template strand it has to be in 3' to 5'. So means the newly formed it is always anti-parallel direction. So because of that, so the template strand it has to be 3' to 5'. Then only the newly formed strand will be 5' to 3'. You see the other template strand, so it is from 3' to 5'. So when it is there like that, so one side it will be formed as continuous strand. So the continuous strand formed you call that as leading strand. So other side it forms in small fragments. So this one you call that as Okazaki fragments. Why is it forming like that? Because so it cannot start from this side. So because here it is 5 dash, so because it has to be, already we studied the newly formed strand is in 5 dash to 3 dash direction. It cannot start from here. So that's why it starts in the opposite direction. It is formed in small bits, then later it joins together. So the small bits you call that as Okazaki fragments, then later it joins together and it forms the strand. You call that as the lagging strand. You can see here. So this one you call that as lagging strand. Okay. So the lagging strand, it is, you see here, this is lagging strand and this is leading strand. So here it is formed in continuous direction, here it is formed in discontinuous way. So that's why sometimes they ask in exam like explain the continuous and discontinuous synthesis of DNA. So why is it like that? Because the newly formed strand is always from 5 dash to 3 dash. So here you can see the helicase, it opens the helical structure. So once it is opened the helical structure, the polymerase it operates. Okay. So before the polymerization operation, so that what happens here? Here you can see helicase enzyme. So it is actually it is opening the helical structure. Then later the RNA primase, it forms the primers. Then later you see one side it forms lagging strand, other side it forms leading strand. So the lagging strand, leading strand you call that as the continuous synthesis, and other side, this side you call that as discontinuous synthesis. So here the DNA polymerase, it elongates. So then ultimately the new strands are formed in both the direction. Okay, then fully completed the DNA, it will be released. So here when you see in the DNA replication, it occurs mainly during the S phase of cell cycle, that is the synthetic phase. In case that actually let us see, for example, one cell it is having 2 chromosomes. At the end of replication, it will double into 4. So then later the cell division occurs. During cell division, first nuclear division and it is followed by cytoplasmic division. So here the nuclear division, when it is not followed by cytoplasmic division, so it results in polyploidy, means the karyokinesis should be followed by cytokinesis. So when there is no cytokinesis, then there is replication result into polyploidy, because the number of chromosome doubles, because 2n into 4n. So after the formation of 4n, there should be cytoplasmic division, then one set 2n will go to one daughter cell and other 2n will go to other daughter cell during growth and regeneration and all, normally during mitosis. So when it does not occur, then it can result in polyploidy. Okay. So already we studied regarding polyploidy, aneuploidy and all. Okay children, this is also one reason for polyploidy. When there is failure of cytokinesis after karyokinesis, it can result in polyploidy. So here what it can be asked in 5 mark question. So you have to study thoroughly. Sometimes they can give the diagram. By giving the diagram, they can ask you explain the diagram or labeling the diagram. In that way also they can ask you in 1 mark or 2 marks, or sometimes it can be asked as bulk question. So explain the DNA replication, like that also it can be asked in exams. So you focus upon this and study thoroughly children. If there is any doubt, you can contact me. Thank you children.